Yeah.
Some giant pry bars in the hangar there that we occasionally take with us.
Some precious coral coming up.
I've been on, we used a bandsaw to help retrieve Herc and Argus.
Wow.
Like
there's a
hydraulic bandsaw.
Sorry.
Oh yeah, go ahead. Sorry.
I saw that flat eel. I thought it was a ray for a second.
Oh yeah, is it an eel?
I see him.
It's kind of a big eel. He looks like he's
cutthroat.
I don't know.
Yeah, can we get lasers?
Elbow macaroni everywhere.
Elbow macaroni everywhere.
This is a very neat feature.
So some viewers want to know why we aren't seeing more animals, which we just saw that great eel, but almost everything that is alive that we're seeing is an animal. We are in complete darkness at these depths. We are at about 1,500 meters, so there are no plants. The corals are animals, the sponges are animals, all the animals. So we occasionally do see fish, and we've been seeing some sea stars and other things. But as far as charismatic megafauna, I guess, we have not been seeing a lot of large super active creatures, although we have been seeing anglerfish and eels. The acrobatic snails were a big hit.
The rocky bottoms aren't going to attract a lot of fish, the types that we see over sediment. But yeah, we've seen some cuskeel-like fish.
This fin is so wild.
Just like really great.
Imagine what this would look like if it was like a mountaintop or something and you were flying this like slowly and close over it.
Our monster truck track.
It's a monster truck arena.
Monster truck arena.
I often imagine snowboarding down these seamounts.
Yeah. So wild.
It's almost cold enough down there to snowboard.
Okay. Go, go, go.
Okay, going. Sorry, got distracted by the
Herc temperature is
the amazing parts.
Around 36 degrees Fahrenheit.
Definitely not a good enough snowboarder though.
There's,
there's another eel.
A nice little eel there.
It's a nice shot.
Yeah.
Another hydrozoid?
Looks like it may be a dead sponge. I can't tell what that is.
I think Gabby's right.
Yeah? Well, it definitely has squat lobsters, right?
Oh yeah, it looks exactly like that thing that we just spent 10 minutes looking very
Hydrozoa.
closely at.
Yep.
Seemed like a lot longer
Same
than
setup.
10 minutes.
Sorry, not sorry.
Yeah, it's pretty.
Time doesn't mean anything anymore.
Speaking of, what am I? Just gonna strafe the gauges there real quick.
Yeah, it's all yours.
Keep your questions coming at nautiluslive.org. You can submit questions in the box there. We'll get to them as we can. Doing our best to get to your questions as we also continue the scientific work that we are attempting to do.
Awesome, thank you.
Is that another Venus flytrap?
Looks
Yeah,
like
it is.
I love them.
Yeah, they're pretty cool. It's not a bad looking anemone.
It's really bent over, is it? Or it's just closed.
Yeah, they always look like that when they're closed.
Zoom video?
Such great textures and colors.
Do you love it?
I love it.
I love it, yeah.
Oh, see, Sarah loves it too.
Yeah.
I want to see another one of those angler fishies.
Me too.
Okay, go
I
on.
really
See?
love.
Thanks.
Any angular rocks here you like the look of? See some chunky monkeys down there.
Yeah, some chunky monkeys. Some of them look very attached, unfortunately.
Oh, yeah. I had auto heading on for a long time there. All of a sudden, I can fly again.
Just think of the happiest thing.
The grouchy anglerfish?
Oh,
No,
that was
'cause
wonderful.
she said I can fly.
Oh, yeah. I love the grouchy anglerfish. Sarah does, too.
I do.
It's hard
Oh.
not to love him.
I know.
That anemone was Pheliactus species.
The big,
Genus
the
Pheliactus.
30, the 30 centimeter one from before?
The Flytrap.
Oh, okay.
Got our glassy seas back. You can see on
Oh,
channel
yeah.
3.
I was actually curious. The ship has been like moving like beautifully consistently. Like I've just been looking at the speed and it's just been like exactly 0.5. And I was wondering if like it was just perfect weather out.
Looks very glassy.
Yeah.
Those glassy seas helped us this morning when a very large oceanic white-tip shark was circling the boat for quite some time.
Oh, really?
Yeah.
That was probably 5 feet at
Yeah, I'd
least.
say 5 feet.
Yeah, Sarah yelled shark, and that was all it took.
Oh
Everybody
my
was at the
gosh.
rail, and it went around and
amazing.
around the ship.
You
Many
guys good
times,
for
many times.
a redo of that move?
It's endangered, the
keep it going.
white-tip
Great,
shark.
Snap.
So it was great to see one alive and well, circling
Can
the
we
ship.
move 100 meters, bearing 225?
Soft coral. And its pom-pom stance. Ooh, another one.
Oops.
Nice Argus shot there.
Watch out for that cliff.
Thanks.
Yeah.
Thanks, Dan. I'm going. I'm going.
Go, go.
Go, 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 go.
So we got some questions about Hercules, who you got a great view there of in SatFeed 2. Is Hercules one of a kind, or is it a production line model that other scientists could get and use?
It's a really long whip.
Yeah.
It's definitely one of a kind.
Herc was built around the year 2000, 2001.
It's a Retigorgia down there.
Yeah.
Pretty big one.
And has been modified and updated and upgraded ever since.
I think the corals like this wall.
Yeah, they do.
If you go on the website, nautiluslive.org, and click on Science and Tech, you can see some really interesting 3D animated models of both Herc and Argus, the two ROVs that we're using today. Just click on Science and Tech and then scroll down.
There's a cool little overhang above Herc.
Yeah.
So we got a biology question. How do these deep water corals and sponges reproduce? How do they know when to release their reproductive materials if they aren't using the sun and the moon like shallow corals?
I don't think
Yeah,
a lot is known about
Don't look
that.
up. Look down and fly up. That blows it out at the top there. Can't see
Okay.
anything. You don't have to look up. You can look up a little bit, but not that far.
Just not till I see the lens flares.
Yeah, yeah. See all the glare now?
Yeah,
So
yeah, I see all the
back- and
backscatter
the
and stuff.
dark spot. Back down just a little.
There, perfect.
Got
I don't know what is known about the timing of them releasing their
Wow.
eggs or the fertilization process. And as Emil said, I don't know if that is well constrained at this time. I imagine Chris might have some things to say about it.
I wouldn't be surprised if in some area where, you know, these internal waves that we've been talking about are the primary source of the current, if they adapt to that
I
somehow.
would, yeah. That was my primary hypothesis as well.
Is that
See
like
that?
a blue colored
Yeah,
one?
the
or is that
purple
just
one
the
down
light?
there.
Oh yeah, that's not often seen, that purple.
Is that a crinoid or a coral?
Let's see if I can get in there.
So those internal waves would be stronger with full moons, new moons, so it could
Okay,
they
go for
could
Yeah.
cue to that.
Wow.
That
It's a
looks
coral.
I
like
think
a
it's
coral,
it's a
yeah.
coral,
I
yeah.
don't think I've ever seen that.
We're getting some input
don't
on
think
this.
I have either.
I think the purple's a giveaway. There's not many that have that color.
Looks like a lavender.
Amethyst.
Amethyst, yeah.
So beautiful.
Okay, change the bearing to 135, please.
Okay,
Sorry,
Nav,
315.
It's an octocoral
Can we get
Oh,
Lee
it's
change
an
your bearing to
octocoral.
type of
315.
octocoral.
Victoria Gorgia is what we're getting from the science chat.
Yep.
Victoria Gorgia is beautiful.
Yeah.
So
Full zoom.
lovely.
The lavender of
Nice.
the deep sea.
First we've seen on our watch.
I'm totally dead
I
stick,
saw
Steve.
that, so that's why I just zoomed, because that's why I looked over.
your
Bearing,
bearing, can you change
Beautiful.
your
Thank
bearing
you.
to 315?
I'm going to try this less is more approach here.
Love it.
Yeah, me too. Okay.
So we're
scram.
getting a question about color. At these depths, it's completely dark.
Yes.
So
coming
why
up for
go
you,
to the
Dan.
trouble to be such a
Yeah.
beautiful color?
We don't know. We don't know if it's a molecule being formed because it has other benefits and that's just a side effect. We do not know why these deep sea organisms would have these vibrant, beautiful colors.
Victor Gorgia.
Sometimes you just want to get dressed up nice.
did
even
he
if you're
punch
not
in
going
the
out.
move, Kate? I'm not
Sometimes
seeing
Oh
it.
yeah, he's yeah,
Sometimes you want to show off your drip.
he just made it.
Steven says sometimes you just want to get fancy.
He was
Even if
getting
you're not
stuck
going out.
without a step.
He's getting what?
He was just without a step. He was kind of pressing a lot of buttons, and like I could see him trying to think of how he wanted to put in change it. So, but he
Well-
put in a 2.25 meters at 315.
Watch what he does and train him up.
Yeah.
So he got confused by the head.
Yeah, he just didn't know.
Maintain
Yeah,
heading, change bearing.
exactly.
You got to train him up a bit.
Here he goes.
Smart guy, he'll get it.
Thank you guys. That purple coral was worth it.
Mm-hmm.
Yeah, I
Good
loved
spot,
it.
Sarah.
Steve helped. I thought it was blue.
What's that? It's not purple? Oh, here's another.
Oh, yeah.
Good eyes.
Wow.
You can't tell me that's not purple. Come on.
It looks
Okay,
purple,
he can
yeah.
resume the 225 bearing.
Is this the same one?
You want to resume the 225 bearing?
Yes, please.
Bridge, nav.
Seems like we have maybe some cup corals
You can call
and
100
Can we
meters if you want.
move 100
the
meters,
Victoria
bearing
Gorgia,
225?
Victor.
Victor
Thank
Gorgia.
you.
Chris corrected
That's
his
the purple
spelling.
one?
Mm-hmm.
Wow. Cool.
Do you love it?
I deeply love it. Do you love it?
I also love it.
Victor Gorgia.
Victor Gorgia is the purple.
Mm-hmm.
Okay.
You're going to
I'll
have
remember-
to hang out for a minute.
The
Alex
ship-
choked there a bit.
Yeah.
Oh, okay.
Yeah, he did some drifting.
Roger that.
He needs to be a little faster on the buttons up there.
Anyone know the name of that yellow one?
Where?
Well, that's great because there's plenty of things to look at.
That looks like a black
Perfect.
coral in the background there.
We
Yeah,
have a
it
new
might
mate on
be.
the bridge, and he's not quite as fast on the button shed as some of the
Can
other
we
mates.
zoom in a little bit on that, Stephen, or is it too hard?
Oh,
We're
yeah,
too bumpy.
We'll get in there. We'll Get in there.
I'm going to go out on a stalk and say that this is my favorite coral we've seen all expedition.
The
Victor Gorgia?
The Victor Gorgia, yeah.
Yes.
Going out on a
You're going out on a stalk.
Yep.
I gotcha.
Yep.
You're going out on a bamboo coral.
That's right.
And is that a big eel right in the foreground?
Oh, yeah.
So Victor Violet. That's how I'm going to remember.
That's right.
Yeah.
Nice.
Oh, that doesn't look like a black coral.
No.
Is that the one that has the zoanthids?
Can I get a
going
video,
on it?
Zoom video? Okay. It's like weirdly in a like a glare and shadowy spot in the light. I'll back up a little. There we go. That's better.
That one was better. He did.
Yeah, that little hook.
Yeah, it should be acute angles there if he's on it.
Beautiful. Color of autumn.
I like that little arm hanging out.
See that beautiful brittle star there? It is not eating the coral. It is a filter feeder. And it's just hanging out, getting higher in the water column so that it can filter its food.
This is a plexarid coral.
Oh, this one is
Off we go.
5 turtles.
5 turtles.
Let's do this.
So we're seeing some species now that were expected in the shallower waters that we're reaching. Shallower is relative, 1490 meters.
I'm going to come back around to 225 there, so.
Copy that.
Light speed.
Yep, I am all ahead. And no Z bias, so really all ahead.
So some differences in the deep water coral spawning. They don't release egg and sperm at the same time the way shallow reef corals do. They brood their larvae in the polyps.
This might be a good rock stop if you see anything chunky and angular. I don't yet, but they don't look like they're attached, so that would make things easier for us.
Yeah, we need to.
Yeah, let me get out ahead real quick and then we
need
can
to
get into that mode.
No worries at all. I don't see any chunky yet, so.
I've been stopping to smell too many flowers.
We got to thank you for the zoom on that plexarid.
Is that the yellow one we just looked at?
Yes, yep.
Too fast. Okay, it looks like we're strangely on the left side of the...
You need to come to your left if we want to maintain that 225, or we can change the bearing to stay up on the ridge.
Croinoid we just went by, a beautiful stalked croinoid.
I bet the ridge will come back around.
Yeah.
No. Let's change this bearing to 180, okay.
Ridge, Nav.
I'm not seeing that in Argus. So.
Can you change your bearing to 180? Come on.
There's
Stalked
another,
crinoid.
another beautiful stalked croinoid.
There you go.
Patrick.
So is he now able to change bearing without a different move?
He moves fast. Yeah.
He's punching him in, but
okay.
I might have messed up there.
I think you're on the spine of the ridge now. This 228 heading looks pretty good.
But, let's change it to ice. I messed up. Should have been 270.
270.
Yeah, I want to go more to the right.
Ridge, Nav.
Slow down, Gabby.
Can we change our bearing to 270?
How big a step is that?
I'm just
We're just
changing.
arranging
We're
Argus.
still making the big step. I'm just zigzagging the ship a little bit to stay on the current side of the ridge.
Gotcha.
So the current is coming from the southeast right now.
That's different.
Yeah, it is different. Or maybe it's yeah, I think it's southeast.
Coming up and over the ridge.
Maybe,
Yeah.
yeah.
It was coming from, I could have sworn it was coming from the west before.
It's very possibly that it reversed.
Looks a little like Star Wars flying into the galaxy here on the HertCam. Sometimes that's called marine snow. What you see drifting there, that's nutrients that our filter feeders are using to filter out. Also seen a couple siphonophores drift by.
Okay, 225 again, please.
Bridge, Nav.
So that's further support for a tidal
Can we
current.
go
Slow down a
100
bit.
meters, 100 meters?
I'm wondering if we shouldn't be
bearing
noting this.
225.
Shouldn't be what?
If we should be noting this.
The current
Yeah, changing
You'll have
current
to slow down,
direction.
Jimmy.
observation.
Roger.
Okay.
I can note that it changed.
Yeah.
Oh yeah, he's moving to the north.
Yeah, not
west.
exactly. 270 move.
I think he's having a hard time with the currents right now.
Yeah, he
He's just having a hard
when
time.
we call the west move, he
Yeah.
he loses it and goes to the north.
Hey, what's...
Don't have a strong wind out there.
Yeah, and if you look at this line cam, it's perfectly flat until he puts on the thrusters.
Yeah, he's the 225 is his heading, so he's just backing the ship down. But when he tries to slide westish, he's
Goes.
so the bow thruster's 90 degrees to the ship, right? In the front. So it tends to push the ship off to port. So he can make an acute turn there when he's drifting northwest to go south again, but he can't do the opposite.
Okay, if anybody spies an angular chunky rock, I spy. You let me know. I don't see one yet.
There's got to be one in this debris field.
Yeah.
You don't see any rocks? I think to your right there's some rocks.
Oh, where?
Big.
If we're gonna get one, we're gonna be quick and go because that's we're moving it half a knot again. Actually 0.7 at the moment. So
Okay, let me know when you feel like it's a good time to drop down.
anytime you see one you want.
Pick him from the top of the screen.
I'm trying there.
Okay,
Yeah.
awesome.
You
Yeah,
can-
I see him. I
You
see
can slow
exactly
down a
what you're
little bit.
talking
You
about.
got
It's like
some
rock
nice there.
Tetris. You
Yeah,
gotta,
yeah,
you gotta act
absolutely.
quick while it's at the top of the screen.
I know exactly which ones you're looking at. I love this that you're keeping up. That's great.
And if that guy is too thin, we got this guy over here. He looks like he might be attached, so then we'll go over here.
I'm going to come to all stop.
This
Okay.
might be the
No, you
guy.
keep it moving. We're gonna touch and go here. That one. Sorry, what was the first one?
Yep, that
Right
one.
there, yeah. He looks
That
like,
one?
whoa, he might be too big, huh?
That's
Yeah.
kind of big.
Okay, our laser's on. I can't see them. There
Yes,
they
they
are.
are.
Okay. Why don't you, yeah, pick up that guy, see how big he is.
Oh yeah, good size.
Yeah, that's a good size. Let's give him a spin, give him a whirl. He's got full beans.
Yeah.
All right, yeah, let's add him to the box that we put the first one in, which is the starboard large bio box in the front.
Roger. Can you?
Thank you.
Can you open? Oh, it is. Thank you.
So a viewer wants to know, why is the rock different colors on the top and on the bottom?
We think that the manganese crust is growing on the top.
And then you can get going.
Also, though, it might also be growing from the bottom. We don't know, but they could be growing at different rates. Also, the top of the rock is exposed to the organisms in the water column. We don't know what effect that's having. And also, it's exposed to the abrasionary effects of moving sediment. So in different parts of the expedition, we've seen that abrasion from what we think is oscillating currents actually will basically sandpaper the rock and make it actually smooth to the touch. So Sarah and I were looking at some of these rocks in the lab, and they look almost glassy and polished, and they're actually very smooth to the touch when you run your hand over them. So those processes are happening at these depths as well. Thank you for getting the rock.
On the fly.
Yeah, going 0.5, going 0.6.
Another crinoid, stalked crinoid.
Yeah,
that's the third one we've seen.
I will get a zoom on the next bright red stalked crinoid. It's got to be out in front a little more.
There's an eel there. Looks like to the left.
Oh, there's a weird little
hole.
Yeah, weird little rift.
Cool.
Are these linear fractures like kind of syn cooling or post cooling, or any idea?
It looks like they might be contemporaneous with the cooling, but I can't tell for sure. They're rounded, right? And it looks like almost like a sheet flow on top of this. Like right, you see that sheet flow right underneath that bamboo coral?
Yeah.
So,
there's another stalked crinoid.
Yes, I promised. I promised myself. Nobody else cared. I really did.
Can you zoom out a bit on high pack, Nev?
Yes, give me 1 second.
stalked crinoid.
Beautiful.
Look at it. It is gorgeous. And it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11.
Go for zoom.
11 feathers? Is that what you guys count?
Oh, I don't know.
Chris says that's important. This one has a lot.
I think there's more like 15.
I counted 15.
Okay, I'm going to keep going.
Oh, you're good.
Yeah, I count 15.
Give him a second there.
Yep, I think it's 15.
You need to zoom out, Gabby?
Nope.
That's beautiful.
Dan gave me the okay to stay.
Okay.
Wonderful.
Now I know he won't give me crap if I get behind.
Beautiful.
You were at the end of your leash there, so.
It is fantastic.
It's gorgeous.
I don't know why I love these so much.
The color.
Okay, now you gotta go.
Okay, I got to go.
I'm going to keep bumping the ship.
Yeah.
Bridge,
Yeah.
nav. Can we
Got an
move
ID.
100 meters, bearing 225?
Okay, thank you.
Stayed in the box though, Gabby.
Rubarimus? Maybe it's because of the ruby
Now you
colored.
gotta make up time.
I know.
That
We're
would
getting a
seem-
system, though. I like it.
It is the system.
It feels like we're seeing more bold colors as we get shallower. And I know there's still no light down here, but
Yeah, that's
I wonder what the
interesting.
connection is.
The tendency towards red is still sort of fascinating me because that's the least visible color you could possibly be.
That's what you want to be, isn't it?
I guess so.
You're hiding.
Keep your questions coming on nautiluslive.org. Thank you so much for joining us here on this dive. I think this might be our 9th dive of this expedition.
Wow, we're already at that many?
Jeepers.
Jeepers. Another beautiful crinoid.
Yeah, there we go.
Ruby.
Yep. Proisocrinus, rubimbarinus.
Is that one little branch coming off of this bamboo?
Oh yeah, just one teeny one.
Talk about sparsely branching.
Walterian sponge.
Those funny ones, the ones that look like covered in fur, those
Yeah.
are called Voltarian?
Walterian sponge.
Walterian?
Yeah.
Okay.
If I'm saying it right.
Slide to your right just a little bit.
To my right, hey?
To your right. There's a
Yeah,
feature there.
Iridicordia.
Oh, feature.
Nice. It's like somebody doctor's signature.
Look at this lovely feature we can follow.
Crack. Yeah.
Good catch, Dan.
Whoa.
Very cool.
Yeah.
So are those layers that we're seeing sheet flows of lava, do you think?
Yeah, it might be all one contemporaneous sheet flow. You can't really tell. And maybe after it cools, it contracts. Or remember that these seamounts are on the order of millions, tens of millions of years old. So they haven't endured some alteration and degradation since they crystallized.
So the dead sponge that fell over?
Where are you looking?
Right below you. Can see
Yes.
his little foot
Oh, yeah.
came away.
Yeah.
Oh, yeah.
He got tall before he fell over.
Yeah.
I guess as a sponge, you have to like not do that, not get too tall.
Yeah.
You have to strategize that sort of thing. Because if you're tall, you eat well.
Yep.
And maybe you grow taller, and then maybe you topple.
Yep.
Zoom a bit there if you want.
On Argus?
Did you want an Argus zoom, Dan?
No, you can zoom.
Oh, me.
Like,
Nope.
yeah.
If you see,
afterburners,
we're seeing here so
Okay.
so
Faster,
little fractures and so little secondary, basically alteration or degradation or slope failure since the formation. And so when you think about this, may have been here for 80 million years, it's really stable over such a long geologic time period.
Come up a bit, just a bit higher, Gabby.
Roger.
That's about right.
There's an eel right in front of us. So we're definitely seeing more eels at this depth. I think we've just seen 3 or 4 in the last maybe 15, 20 minutes.
I'm going to bump your camera.
When you get a chance, could we get a zoom out on high pack?
Wow.
Oh.
Is that a
cool.
different eel? That's
His
not
head
an
is
eel.
different.
I don't know what
No.
that is. I don't think we've seen that before.
Yeah, the head looks
Okay.
different.
The
Go
head
ahead.
looks
Go,
different.
go.
Gotta go.
What do we have? Are we doing 100-meter steps, Nev?
Yes, we are.
Looks like a good line.
Yeah.
So on feed 3, you can see our
Make
high
up
pack.
ground.
If you're interested in,
What's that, Dan?
You're going to make up ground?
Yep.
Faster, faster.
if you're interested in depth and other information, got a couple of ways of accessing that. You can go to the website nautiluslive.org, and you can see our current depth as well as the temperature right there on our homepage.
It's getting steeper, so you got to go faster. Yeah.
Oh, really? 626.
Where are you going? Yeah, they're happy with this line. I'm happy with it.
We are
Keep
getting
moving.
to a steeper area.
We are.
The contours are getting closer together.
Yep.
Do another move.
I'm looking at Argus, Gabby, so I think we're on a good line. And the high pack, what's the way we want to go?
Bridge, nav. Can we move 100 meters, bearing 225?
It's unbelievable how still the water is on the line cam. Now as he puts the thrusters on, you can see it change, but
You probably got time to pirouette all the way around that rock if you get on with it.
Another great question from a viewer. If this seamount is 80 million years old or so, why is there so little sediment?
well, there's so little sediment because one reason is we're far from a continental setting, which will usually be the primary source for clastic input. And so we're out in the deep, deep ocean and far from a continental source, number one. So the sediment that you would see here would be largely derived from organisms. And so those organisms might be in the photic zone at the top of the ocean, where they are creating tiny little tests or shells as they float through the water column. Those might be formed of siliceous material or calcium carbonate.
Siliceous material?
Silica-based material or calcium carbonate. Or you might have, what we see here is that sometimes there are barnacles on the rocks at this depth, and their shells are actually making up the sediment. So those are two of the reasons that you have so little sediment out here. In the pelagic zone, the sediment is so sparse that you can actually see in sediment cores taken in these areas the wind-blown sediment that comes off the terrestrial areas,
coming back to 225.
Even off some of the deserts, and it can travel from continent to continent, and it's so fine it gets tossed up and sucked up into the higher parts of the atmosphere, and it actually will rain down in the abyssal zone and travel through the water column. There's actually also such little sediment input in the abyssal area that you can measure galactic dust that travels to our planet from other parts of the solar system and deposits and can be analyzed for its helium-3 content.
You're under me now, so you're going to want to go 225 and go light speed. Roger.
We do seem to see sediment accumulating in certain areas, and that would be the influence of currents, and we're especially interested in looking at large areas of sediment to see if we can see ripples and evidence of these tidal-driven currents at these depths. And so some areas are completely free of any sediment at all due to the action of currents, we think. Also, all those corals and sponges gobbling it all up, right?
Yeah, they're definitely using the pelagic snow and as a source of nutrition to build these
Gonna get steep now.
Beautiful organisms that we see.
Yeah, if it does get steep, you won't have time to stop at this speed. You'll have to. It's going to be a handful to go forward and come up.
Another beautiful stalked crinoid. So those are fairly abundant in this area. The Rubarius,
Seen a couple
Rubarimus, yeah.
Couple more of those Victor gorges as well.
They are beautiful. Kim loves them.
I do.
doesn't look that steep.
Looks like another Venus flytrap, potentially. That one was open, and an eel.
Thanks to everyone who's tuning in on our website. We have viewers from around the world right now tuning
nice
in.
eel. Looks very similar to the one we did a zoom in on before.
Yeah.
Those ones have like pointy noses.
Yeah.
It looks like a seahorse head.
Yeah.
We're hearing snailfish or halosaur as a ID on that pointy-nosed eel-looking thing.
Halosaur sounds like a dinosaur.
I know.
I like it. It's a lot of those crinoids.
Yes, they're beautiful. 2 in this shot.
Yeah.
Emil, I'm not sure if you've been looking at the HYPACK screen, but we are at this speed 20 minutes from the end point.
Yeah, we would continue on
Okay,
past the
copy.
end point.
Abyss?
Because we are hoping to get past that endpoint, is that correct?
Bridge now.
Yeah, if we
To get to the summit.
we'll pause for any interesting communities, but yeah, we'll.
Can we move 100 meters, bearing 225?
Might start seeing the transition of the carbonate cap.
Yeah, that's what I've been looking out for, wondering if we might hit it on our shift here.
Unless there's
excited to
something
see it.
interesting, I try and stay almost to the point where I'm tugging Argus the whole time. That gives you that much more, 5 seconds or 10 seconds to look at something if you want.
Oh, was that trash?
Yeah, that was like a Modelo can or something.
A Modelo can.
Another little eel there. Ooh, I just can't.
Wow.
They're
Wow.
so
Pretty.
fantastic.
That one looks like more than 15.
It does.
Brilliant.
Brilliant.
I'm not going to try to count.
It's been a colorful dive
Yes,
between
it
that
has.
hydrozoa, the Victor Gorgia, and these lilies.
We're about halfway through this watch. We'll be handing off to our counterparts on the next watch, so the exploration will continue. We got a young viewer in Connecticut wanting to know, if there's no light at this depth, how deep does the light penetrate in the ocean?
For photosynthesis, only 50 to 100 meters. It depends on water clarity. A couple 100 meters at the most, but you'll have some bits of light down to about 1,000 meters. Below that, you're in the dysphotic zone.
The twilight zone is to 1,000 meters, is it?
Twilight's like the depth from the bottom of the euphotic zone where the photosynthesis is going on to, yeah, down to about 1,000.
Great question. I would have thought it was much shallower.
It's not light that we can see, but it's light that,
It's measurable.
yeah.
Sure. Thanks so much for your questions. We love getting questions from those young viewers out there. We're right now at a depth of about 1,340 meters, so below that final depth where any light can penetrate.
So do we usually have more weekend viewers or more weekday viewers? I always wondered.
I think the main difference seems to be what time it is where, I know this is a rare daytime watch for your 12 to 4 crew. Usually we're midnight to 4 a.m., but it's a rare noon to 4 p.m. watch for us. So thanks for tuning in from wherever you are, whether it's good morning, good afternoon, or good night where you are. And keep those questions coming, and we'll get to them as we can.
So if we are approaching the carbonate shelf, we do want to get another angular rock. I know we're on the fly, so whenever you get an opportunity to slow down, we can pick up another rock.
We can't hear you.
You're not on SPL, Gabby.
Yeah, so I'll just come up a bit higher so you can see more.
Okay.
And then we'll do that Tetris game again where you pick something from the top of the screen.
Okay.
We'll scream in and grab it.
All right.
At a screaming speed of 1.5 turtles.
I mean, it sounds so smooth.
It's all relative. It's all relative.
Dan, was there something else you wanted to do
We've
instead?
been pretty constant at a 10 meter a minute ascent rate. So we're going forward at half a knot and coming up at 10 meters a minute. Why we don't use meters a minute or meters a second for vessel speed, I don't know. But
half a knot is 0.26 meters a second.
Is that a cucumber?
Yes.
or 50 centimeters a second.
Yeah.
Not many ships use that.
So we're only 2 hours in? I thought we were...
Yeah, a little bit more. We're 2 hours and 15 minutes into
What
our
do you see here,
watch.
Andrea?
Sorry, I got pulled away for strategy. Let's see. You see anybody see anything angular in there? That is not attached. Maybe this guy over here?
Yeah, sorry, those ones that just went out the bottom.
Ooh.
Okay.
I got to get out ahead again. Sorry,
Okay.
a little bit too close.
No
Sorry.
worries at
Yeah,
all.
stretch it.
I was
stretch it
trying to get you in a new vein of crushed sort of debris and got behind.
So we got some questions about how we're processing these rock samples once they get here. If you tune in after a dive, the live stream continues. We don't answer questions, or we're not on SPL at that time, but you can watch on nautiluslive.org. We often have a live shot
You're
up
still
of our
under
lab.
Argus there now.
We have a lab here
What's
on
that?
the ship.
You're still under Argus. Got a
It
ways
doesn't
to
look
go.
like it from fisheye, though,
I
right?
can see it.
Okay, let me know when I should have my eyes peeled.
But your camera's way forward.
Yeah, the top left one's looking straight down, isn't it?
The top
no.
left one. The top left one is fisheye, and it does look straight down, but it
Yeah.
Whoa, what is that?
Yeah, what
It's
was
a
that
rock.
spiky thing?
Oh, yeah.
And we have
It's a
the
rock.
Victor Gorgia.
It's a rock.
It's a rock that I almost hit.
You're starting to sound like Andrea. That rock, I love it. So we got a question about trash. What's the protocol when we see trash? Do we ever pick it up?
Yeah,
Or
we do.
It's time for another move. Do we want to
Oh, there's
settle
a
out for
dirty little
second,
fish.
or keep on
Yeah,
moving?
he's fast. He looked like a gulper eel, kind
We
of.
can if we want to get a rock.
Okay.
Andrea, do you want to slow the operation down a little bit to get a
Yeah,
rock?
do you see anything there?
I'm still not out far enough in front, unfortunately.
Okay, you let me know when you're ready.
So we can stop the ship
Ship is stopped.
Yeah.
Okay. Yeah, we can. Yeah, let's just leave it stop for a little bit.
That sounds like a good idea.
I haven't stopped to look at anything in a while. I'm still not catching up.
Well, you're coming a little steeper now, so you're...
We are getting steeper. This is a good time to stop.
Coming up 12 to 15 meters a minute.
That looks...
These guys are good over here.
Okay, keep an eye out for more of those, but I think we're still working at settling everything out.
Yeah.
Oh, that one's a nice one. Nice chunky guy.
Don't say that, she can't grab it.
Can we get lasers on, please?
So.
Was that a lasers on sound effect?
Pew,
Is that what I heard?
Pew, pew.
Yes.
So we are operating in Papahanaumokuakea Marine Monument. So our operating permit asks us to geotag any debris or trash that we see.
Big sponge at it.
We've seen very little on this expedition, just a couple of cans. So we tag that so that if they wish to come and remove any larger items, that they can do so in the future. But we are not picking up any debris that we see on this expedition, because that is outside of our operating permit in the Marine Monument.
So just talking to our lead scientist, or co-lead scientist, Chris Kelly, we're going to extend this dive till midnight today, and we will explore part of the summit, see what we got up there, maybe explore along the rim of the summit.
Oh, there's a good rock. There's
That's
a nice
good
chunky
rock.
monkey.
Okay.
And it'll be interesting. It'll be a different sort of habitat, the top of a keel. It's very angular. There you go.
You're not on SPL, Andrea?
Sometimes I think it's better I'm not on SPL.
No, we must hear you say.
That's what
no.
Gabby says.
Okay, off you go.
We must hear you proclaim your love.
That looks beautiful.
Oh,
looks
that
great.
one looks like a chunky monkey.
Just grab and go right there.
Yeah.
So you really like the sharp angles there on
Yeah,
that
it gives me, I think it's a good chance it's crystalline through. Is that giving it full beans?
It is, yeah.
Uh-huh. That looks like a good one. We can put him potentially in a starboard bio box that is not the one we've already used, so I think the other bigger one.
Yeah, Roger.
So will you be breaking this rock down for analysis on the ship? And if so, are you using
Go ahead.
saws or hammers?
No, we will break it down once we get to shore, and we'll get it in the lab, and we will saw it open. Then we will look and find the freshest parts of the rock. We will identify them, put them in a rock crusher. Then we will sieve them and put them in a hydrosonic bath with a millicu water and clean them. And then we will put them in the magnetic separator to target the minerals that we want. Then we will sieve them again. Then we will put them in a leaching process where they will be leached with 3 different types of acid over a day.
There.
And then we will put them under a binocular microscope where we hand pick the best minerals, which we refer to as our target phase. We will select them until we have enough mass to be measured. Then we will send them off to a nuclear reactor where they will be irradiated. And then after they cool, they will come back to the lab, and we will load them in these foil capsules and into a glass, take them out. They're already in these little foil capsules, and take them and load them into the machine and measure them with what we call incremental heating steps. Release the gas through different phases of being heated to higher and higher temperatures to release the gas to analyze it to try to determine the age of the rock. So there's a lot involved in preparing these rocks to tell their story and tell us how old they are. But when we get a good age, we are really happy because it is very valuable in understanding how the Pacific plate moves over geologic time and the relationship of these seamounts to potential plumes responsible for their formation.
Oh, look at that radial pattern there on that rock. Looks like a nice one. So
Are you moving again, Kate?
Nope.
we are getting some questions about
You ready, Gummy?
For a move?
how we
Not
know
quite yet.
where we selected each rock from. That might be a Kate question.
Or a Sarah question. Sarah, do we have lat longs automatically populate the fields when we select a sample?
Yes, every time I hit sample submit on my event log software, it will automatically tag a lat long and all the other metadata that you would want to know about a sample. And then the navigator will also drop a location into HiPAC.
Nice.
Also baked into our video is lat long, dive number, and depth, and it's kind of like closed captions for a movie, or subtitles. You
So
can
we
toggle them on
always-
and off in the raw video.
So we always know where we are.
Hey, Nav, could we zoom out a bit?
Bio break there.
Yep.
Sorry, zoom out.
Yes.
Yeah, give me
I
one
want to see
second.
the summit.
I'll keep it stopped until you're back.
So Andrew, what's the age range you would expect? Because I would imagine you could get several rocks on one seamount and have several ages depending on the flow timing, like when it erupted.
Exactly. So an underwater seamount or any volcano does not form instantaneously.
Let's drop a target. The beginning of keep going a little bit further with your cursor.
Sorry,
Yeah, right.
I've got my eyes on other things right now, so just a little patience.
That's also why we hold the rock
Where would
up
you like to
to the
target?
camera.
A little bit closer to the edge. A little bit northwest, right about there. And let's drop a target there. We'll proceed up there.
What would you like the target to be named?
Edge of summit.
You got it.
Is that a cup coral?
The baby.
It's bigger than any cup coral we've seen, I think,
Uh-huh.
if it is.
So we get a really nice view of each rock that we collect with the cameras. So if they are in the same collection box, we can tell them apart visually. And then each sample is tied to that precise location.
It's a single polyp sclerotian or cup coral.
Yeah.
Can you finish your story?
Oh, yes, the age. Yes, the age of a seamount. It takes time for a seamount to build, for a volcano to build. So the rock we get will just be essentially from one flow or one portion of the event building the seamount. And so you can get a range of ages, but with every age you have an error, meaning if this rock is 80 million years old, it might be plus or minus a million years. So the entire age is 80 million plus or minus 1 million, meaning that age is 79 million to 81 million. So when we get multiple dates on multiple rocks from one seamount, they usually generally fall within error of each other because we're going so far back in time.
Okay, okay.
As we get younger rocks, so for instance, I have dated rocks from the big island of Tahiti, Tahiti, and they had errors of only 3,000 years because they were only 900,000 years old.
Okay.
So what was the error on your 70 to 90 million year old?
She said up to 1 million or more.
Oh, wow.
Yeah.
Yeah, it's been off. It's hard to imagine Argus in front of the ship unless there's a whole lot of current.
So it is easier to date younger rocks or easier to date older rocks?
It depends on how fresh the rock is, how unaltered it is. Generally, because you need some ingrowth of the argon,
Excuse me,
Yeah.